be used to produce ATP. So as I've mentioned, the liver is an important fuel source during exercise. So we do have carbohydrate stores within muscle that we can use, and also rely on both glycogen stores within our liver and its ability to produce glucose via gluconeogenesis to sustain the demands of exercise. We'll look at some data from some healthy individuals and also people with cirrhosis to understand the importance of fuel provision from the liver. The disease in which liver tissue is replaced with fibrous tissue, which decreases its function. It's associated with alcoholism and hepatitis. It's a fatty liver disease. We have a fat deposition in the liver that prevents it from performing its normal function. So what we'll look at is what happens to the ability of the liver to produce glucose under this disease state, and what happens to the ability of people with cirrhosis to exercise. So this graph here is showing you plasma glucose. We rely on plasma glucose during exercise as a source of substrate for glycolysis to produce ATP in our muscles during exercise. So what we see in individuals with liver disease, in this case, it's represented by the open circles, both as rest and during exercise, we have less glucose available in the blood. So this is happening because inflow of glucose from the liver is impaired in cirrhosis. So this leads to an impaired ability to exercise because they can't provide their muscle with glucose to produce ATP. So muscles cannot generate as much force as they would in a healthy individual. Other thing we see in individuals with liver disease is that their livers excrete a lot of fats, and sort of a compensatory mechanism to provide the system with the energy since glucose production is impaired. And that results in elevated fatty acids concentrations in our blood, both at rest and during exercise. And during exercise, it's good because muscles can use these fatty acids to produce ATP. And at rest, this is bad because elevated fatty acids in our blood are taken up by many important organs or tissues in our body and impair their function. So that's it for our gastrointestinal tract and liver. So now we're going to move on to the next source of substrate, which is our adipose tissue. So we're going to look at the role of adipose tissue in fat storage and release. And adipose tissue is all of our fat cells or adipocytes within our body. If you look at an adipocyte or fat cell, it has similar organelles to most cells. It has nucleus, mitochondria, also has a large triglycerides droplet. It makes up 80 to 95% of the adipocyte mass. And as we gain weight, what happens is that this triglyceride gets bigger, so our adipocyte expands. So when this happens across many adipocytes, we get an increase in fat mass. So the major function of the adipose tissue is take up and store fatty acid from blood or release fatty acids into blood. And like the liver, it has the ability to synthesize fatty acids from carbohydrates, but doesn't happen very much in adipose tissue. Most fatty acid synthesis occurs in liver. So the adipose tissue primarily is a storage site for fatty acids for when they are needed. And in terms of uptake and storage of fat, what happens in the adipose tissue is a process called re-esterification, where carbohydrates or glucose are taken up into adipocyte as well as free fatty acids. So the glucose is broken down through glycolysis. It produces acetyl-CoA. The acetyl-CoA activates the fatty acid and turns it into a fatty acyl-CoA. In addition to this acetyl-CoA, we have glycerol being produced from the breakdown of glucose. And the activated fatty acid CoA combine with the glycerol to form triglycerides. And the entire process is known as re-esterification. So in the adipose tissue, fatty acids are taken up, re-esterified into triglycerides, which are then stored in that adipocyte. In addition to the ability to re-esterification and store of triglycerides, the adipose tissue also has a capacity to break down stored triglycerides molecules. Remember that a triglyceride is a free fatty acid combined with a glycerol backbone. And we have the lipases, which cleave off free fatty acids from the glycerol backbone. So the free fatty acids are then excreted from our adipose tissue into blood, where they can be delivered to areas where they are needed the most. So this is what lipolysis looks like in more detail. We have our triglyceride, which is a glycerol backbone with 3 fatty acids. And then the lipases cleave off individual fatty acids from that glycerol backbone. And eventually we have 3 fatty acids and glycerol. And all of these are released into blood. So importantly, in our adipose tissue, we have three different lipases. One that cleaves free fatty acids from triglyceride, one that cleaves free fatty acids from diglycerides, and one that cleaves free fatty acids from monoglycerol. So we have one that cleaves the free fatty acids from triglyceride molecule, another one from that cleaves from the diglycerol, and another one from the monoglycerol. Okay, so finally, we look at this. We already look at this when we look at metabolism, but when lipases produce free fatty acids in our adipose tissue, in our adipose cells, they are released into blood and taken up by cells that use them to produce ATP. For instance, skeletal muscle. So transporters on our muscle membrane bring these fatty acids in. They are subsequently transported and activated by CPT1 and then metabolized via beta oxidation. And this is an important source of substrate. So when we fast or when we exercise, the longer we go without food or continue exercising, the greater the reliance on fat becomes. Both during fasting and exercise, when we're not taking in a lot of exogenous fuels, where we're not ingesting and digesting and absorbing carbohydrates, fats from food, we rely really heavily on the release of free fatty acids from the adipose tissue. So these large stores of triglycerides in adipose tissue start undergoing lipolysis. Free fatty acids starts being released into the blood, and our tissues take up these free fatty acids and start using to produce ATP. Okay, so as I mentioned, we have these different lipases that break down triglycerides, diglycerol, or monoglyceride. So the adipose triglyceride lipase, also known as ATGL, cleaves the triglyceride. The hormone-sensitive lipase, also known as HSL, cleaves the diglycerol, and the monoglyceride lipase, also known as MGL, cleaves off the monoglycerol. So this is just to show you the importance of adipose tissue during exercise. We're going to look at a study where researchers generate mice where they deleted this ATGL from their adipocytes. Therefore, those animals couldn't start lipolysis. It cannot produce free fatty acids to provide muscles with free fatty acids during exercise. Let's take a look in this study. So they had wild type mice, and they had another group with ATGL-KO mice. So those ATGL-KO mice are knockout mice for this lipase. So these mice have their ATGL enzyme deleted. So in the wild type mice, we see that with exercise, we have a larger increase in plasma free fatty acids, meaning that we have lipolysis going on in the adipose tissue and free fatty acid being released into the blood. In the other hand, in the ATGL knockout mice group, it cannot initiate lipolysis. So there is no change in plasma free fatty acid concentration. So under normal conditions, we have fatty acids being released from adipose tissue into our bloodstream. And in this knockout mice, due to the deletion of that first lipase, they cannot undergo lipolysis. So there is no fatty acid breakdown and release. And if we look at how this impacts exercise capacity, we see that our wild type animals have a high maximal running capacity and endurance capacity. So in our ATGL knockout mice, we see that both their maximum running capacity and their endurance capacity is impaired. So what this shows you is the importance of fatty acids from adipose tissue being provided to our muscles during exercise. If we didn't have that ability to break down fatty acid through lipolysis and deliver them to muscle to produce ATP, we wouldn't be able to exercise as well as we can. So the same thing happens for fasting or starvation. If we didn't have these fat stores that are our source of energy, when other sources such as carbohydrates are depleted, there would be no way for our cells to produce ATP. So that's it for our sources of substrate. So in this slide, you're gonna find a summary of what we went through this lecture.